All right, thanks. Thanks for that. So thanks to all other speakers today. It's been some awesome talks so far. My name's Chris, and I'm Justin Brooks' honours student, and I was co-supervised by Virginia Gaget. So before I start, I'd just like to acknowledge the traditional owners of the site in which I studied, the Coorong, so the Nurrindjeri people. And while the site is extremely ecologically important, as established earlier by Claire, it's also extremely culturally important and has been through many generations for the Nurrindjeri people. So today I'd like to talk to you about potential recovery avenues of research through making sense of microbial communities and their functional roles in extreme environments such as the Coorong. So first of all, what is an extreme environment? It could be anything from an acidic crater lake in a volcano, a really cold environment like this ice core, a warm environment like a geothermal spring, or hypersaline lakes like Lake Hillier in Western Australia, or hypersaline bodies of water like the Coorong. So you don't think of these environments as being particularly conducive to life, but they're actually host to a whole range of organisms that we term extremophiles. And these organisms undergo adaptive measures. For example, in hypersaline bodies of water, organisms will perform osmoregulation to balance the solute concentration intracellularly with the environment. So you can think of it as a bit of a matter of perspective. The anthropogenic lens sort of makes us think of these environments as extreme, but in some cases there are organisms that require these specific conditions to grow. So I'd just like to separate extremophiles from extremotolerant organisms. So there are some organisms that are tolerant to these conditions, and then there are others which thrive and require these conditions for growth. So why might we study these microbes? Why are they important? They help teach us about adaptations of life to harsh conditions, the origins of life, astrobiology, which is super interesting. It involves the study of extreme environments on Earth and extrapolation to potential life in extraterrestrial environments, and biogeochemical cycling. And I'm particularly interested in nitrogen cycling. So this is the transformation of various elements through biotic and abiotic factors within the environment. So how do we study these organisms? Here's one example. This is my lab mate Tyler, and he's holding the sampling device that we used. So we screw cores into that, and then we can suction up little cores of sediment and then analyze what's present in those. So as for the study site, the Coorong Lagoon, it was recognized as an internationally important wetland under the Ramsar Convention in 1985. And this is because it's a really important habitat, as Claire was talking about, for migratory waders. And it's also an important habitat for a lot of threatened freshwater fish. Sorry, not threatened fish species. So it's present at the estuary of the Murray-Darling Basin, which is the largest river system in Australia. It drains most of the southeastern quadrant of the continent. And it's controlled by freshwater flows from the Murray at the north through a system of 5 barrages, which were installed in the early 20th century to prevent salinization when we started developing agriculturally upstream. And also tidal exchange through the Murray mouth with the Southern Ocean. So it's separated into the Northern Lagoon and the Southern Lagoon, tenuously connected at Pinker Point. So this point sort of acts as a bit of a hydrological choke. There's also some freshwater input at Salt Creek through the Morella Basin in the south. And because of the nature of this, it's host to a salinity gradient. So as salt water comes through the mouth, water evaporates and solute accumulates further up the Coorong. So what you get is a salinity gradient. And in the southeast, we have quite high salinity. And then it graduates down to marine salinity in the Northern Lagoon. The Coorong's been party to degradation, much degradation and drought throughout the years due to over-extraction, the Millennium Drought, for example. And also the freshwater flows through the North and South Lagoons are both from high agriculturally developed areas. So they carry a lot of nutrients generally. And this can lead to algae growth. And we've seen that in the South Lagoon. So here's a photo taken by Dave Payton in January of 2017. And you can see these algal mats covering much of where the migratory waders would be feeding. So it's interfering directly with the habitat of these birds. And it's also interfering with the growth of other endemic species which are important to the system, such as the aquatic macrophyte Rupia tuberosa. So my research questions for the honours project were as follows. How do the Coorong's hypersaline conditions affect microbial community structure and diversity? And how do other physicochemical variables affect this structure and diversity, such as nutrients, dissolved oxygen, and water depth? And then how do all these changes go on to then affect microbial functional roles, specifically denitrification? So what is denitrification? It's the reduction of bioavailable nitrogen forms able to be used by the autotrophic algae to unavailable forms of atmospheric gas, which can't be used by these organisms specifically. And it's performed by a diverse range of microbes, mostly in the absence of oxygen. So instead of using oxygen as a terminal electron acceptor, nitrate, nitrite, and so on are used as the electron acceptor instead. It's important in estuaries in limiting nitrogen. In a healthy estuary, this process contributes to nitrogen limitation. And for this study, I looked specifically at the gene NeoK, which codes for the copper-containing nitrite reductase. And the reason I chose this step is it represents the first committed step to denitrification. There are other parts in the process that feed into other various processes. So this one represents the committed step to denitrification, and it also results in the first gaseous intermediate, nitric oxide. So for sampling campaigns, I had 2 separate sampling campaigns at the end of last year. So I sampled for the preliminary Jack Point and Long Point, which are about halfway through each lagoon respectively. And we used the boat to go out and assess different depths to have a look at the tidal influence on sediment focusing. So to have a look at the differences in communities between that. And each depth was taken in triplicate. And then for the primary sampling campaign, I sampled along the salinity gradient from the highest salinity site, Salt Creek, up to the sort of marine salinity level sediments up here at 2 different depths for each site, and they were each sampled in triplicate as well. So the molecular tools I used, first off, I extracted DNA and RNA from each of these, and I used quantitative PCR to measure absolute gene abundance against a standard curve. And then calculated cell abundance and did the same with the RNA with reverse transcriptase, reverse transcription, how to look at the transcript abundance against the same standard curve. This was done in parallel to next generation sequencing of 16S rRNA, as Ella was just talking about, the same region, V3, V4, which is a sort of barcode for prokaryotic microbes. So the extracted DNA, the 16S gene is amplified and then sequenced. And then I use various bioinformatics platforms to sort of learn about the bacterial and archaeal community composition parallel to these. So for results, I found that salinity was the main determinant of overall sediment community structure. And you can see that this effect occurs along the salinity gradient on this correspondence analysis here. This is an ordination type that's useful for detecting ecological gradients. And you can also see on this plot, this bar plot of relative abundance of major phyla over 1%, that there's a shift. So at hypersaline habitat, they were much more archaea, and then they sort of drop off as you get to the lower salinity levels where cyanobacteria dominates a little bit more. And in addition to this, alpha diversity was slightly reduced at Salt Creek. Yeah. Okay, and to have a look at the other physicochemical variables and how they might influence community diversity, I use a constrained correspondence analysis. So you can see that there's a clear separation between the 2 lagoons. Total nitrogen was elevated in the Northern Lagoon with NeoK cell abundance. These were both highest in sites in the North Lagoon, and then total phosphorus was elevated in sites in the South Lagoon. Interestingly, although cell abundance was elevated in the North Lagoon and dropped off with higher salinity, activity was higher at Salt Creek, which is the most saline environment. And you can also see on this taxa plot that there's a big difference in community composition. So you can see the archaea sort of are present here and they're not present at all in the Northern Lagoon. And the same goes in inverse for cyanobacteria. So there's definitely a community composition shift, and there's some interesting results with the abundance and activity of near K. So to have a look at why this effect might be occurring, I had a look at the significant taxa across different salinity levels. So samples were separated into hypersaline, euhaline, and polyhaline. And I found that 4 of the top 10 significant hypersaline taxa were archaea, which might indicate that these taxa are contributing to the increased near K activity that we saw. In the lower salinity habitat, less than half of the significant taxa were identified. So next, I put my 16S data into an algorithm which allows us to infer the metagenome from 16S sequences by matching taxa to the closest relative within databases. So the closest relative with a fully sequenced genome. So there are some limitations inherent to this, just purely because of the fact that these databases are going to be limited by the previous studies that are done. So there's not going to be, for some of the taxa that are present in harsh environments, there's not going to be fully sequenced genomes. So the closest relative might not be wholly representative of that that taxon that we're in particular. So this data sort of is suggestive in nature and allows us to formulate future hypotheses rather than actually drawing specific conclusions. And I saw it in this data, I saw a shift from high metabolic plasticity at low salinity. So there's a lot of different metabolic pathways that were elevated. And this is likely due to the freshwater inflow, which is highly modified, highly anthropogenically modified. And then in higher salinities, pathways were related to cell growth and transport, which is indicative of osmoregulation. So of the extremophile taxa in the hypersaline sediment. And nitrogen metabolism itself wasn't inferred to be much different between the different salinity levels. But when you think about the context of nitrogen metabolism as a whole, there's a lot of competing pathways and a lot of different things that are going on. So to have a look a little bit closer at that, I had a look at the inferred orthologs specific to denitrification. So inferred near K abundance was similar to the actual empirical abundance measured by quantitative PCR. And the rest of this data also suggests future hypotheses for denitrification study in the area. Sorry, my mouth's gone dry. So to have a look at the specific taxa and dominant taxa that were inferred to be harboring near K, I had a look at relative abundance on a heat map. And so this is the northern side, and this is the southern side here, and these are the different depths. And you can see that the dominant taxa that are inferred to harbor near K in the Northern Lagoon are sort of marine bacteria, such as Rhodobacteriaceae, which is a metabolically active family, which is poorly characterized. And then in the Southern Lagoon, I found that this genus of the family Thermodesulfovibrio was dominant, and it's a strict anaerobe capable of both nitrogen and sulfur reduction, as well as some other halophilic bacteria and archaeal taxa. So in conclusion, salinity shifts community diversity and negatively impacts near K denitrifiers. But near K denitrifiers are more active in hypersaline habitat. Alpha diversity was lowered at Salt Creek, which was the most saline site and had the highest near K activity. This could indicate niche adaptation and specialization of the taxa that are performing denitrification in this area. And as for the inferred metagenome results, the functional role shift from high metabolic plasticity in low salinity to more growth and osmoregulation dominant functional roles in higher salinity was suggested, not concluded. So in future directions, I'd like to look at time series analysis alongside hydrological modeling, because nutrient delivery and just the Coorong itself is highly complex in nature. And I'd like to expand the assay genes to widen the scope and also improve robustness of the metabolic inference with parallel shotgun sequencing or just direct shotgun sequencing of microbial communities. I originally planned to do a whole range of different denitrifying genes, but due to some issues with getting the assays developed and that sort of thing, I sort of ran out of time, and then that's when I chose to do the algorithmic inference instead. All right, so thanks for listening. Were there any questions at all?
Thanks, Chris.
Cheers.
That was really good. I'm just going to pull this up. Right, anyone raising their hand that want to ask a question? No, I can't see anything at the moment. Oh, yep, Frank, you'll have to just turn your audio on.
Thanks, Chris. Really interesting. So has a similar study been done in a similar scenario somewhere else to see if that's something that's observed in other places as well? I mean, your correlation with salinity and denitrification, is that something you think that's going to be the case in other scenarios as well, or you think that's more a local phenomenon?
So it's thought to, the literature is kind of, it's kind of conflicting. There's some papers that would, indicate that it's increased in high salinity, and some papers that would indicate the opposite. But there's also, I'm not sure if I mentioned it, there's also another gene which is kind of functionally redundant to a certain degree. It's a different form of nitrite reductase. This is another gene assay that I was trying to look at. And there are theories that these two play different roles in different salinity environments because they have different reduction potentials just due to the nature of the enzyme itself. So yeah. Did I answer your question?
Yep.
All right, cool.
Okay. Hey, Chris, what sort of modifications might you think about doing to the Coorong to increase denitrification rates? Or are there any modifications you could do based on your results?
So obviously, this is just a slice in time. So it's not indicative of, it doesn't take into account seasonal variation or anything like that. But I don't think there's much you could do to modify it, like directly modify it, but it might help to inform like freshwater releases and that sort of thing.
Okay.
So through the Morella Basin, there's like a high nutrient load that's coming through that water. So it could help to inform maybe treatment of that water before it gets there or something related to that.
Actually remove the nitrogen in the first place.
Yeah, well, yeah.
David, did you have your hand up?
Did, and Chris, you partly answered, and that is that the salinity changes seasonally in most places there by maybe 30 grams per litre.
Yeah.
How does that change the likelihood of denitrification varying across the season and you not actually having something which is representative of that system as a whole?
Yeah, so, well, that would be the next step to have a look at that, obviously. But I think it's likely that salinity is pretty tightly coupled with denitrification. So it's going to vary seasonally as well. And then, yeah, so it's complex, and it definitely requires a time series analysis to have a look at that specifically.
So is that just going to be a change in the organisms where they're active, or change in the actual composition of the organisms which are present at sites?
So I think there was some of the evidence did sort of suggest, from what I was reading, did sort of suggest that the community in the hypersaline sediment is quite well established. So I'm not sure whether there would be like a great deal of variation, but just the actual nutrient delivery and that sort of thing could have heavy consequences on it. There would be some change. There's likely to be some change in community composition seasonally.
Okay.
It would be interesting to look at. It would be the next step.
Thank you.
Right. Thanks, Chris.
No worries. Thanks for listening, everyone.
If you could just unshare your screen for me, please.